Hello and welcome back to the Sleepy Darwin. Whether you're here to let your mind drift into peaceful sleep, to unwind after the complexities of the day, or to simply ponder the quiet mysteries that surround us, you've found exactly the right place. Tonight, we're embarking on a journey into the most intimate instructions of life itself, those 4 simple letters that spell out everything from the color of your eyes to the rhythm of your heartbeat. We're exploring what happens when humanity learns to edit the code of life, when molecular scissors borrowed from ancient bacteria become tools for healing, and when the alphabet of existence becomes something we can read, understand, and gently rewrite. Before we begin this soft descent into the world of genetic editing, I'd be deeply grateful if you could tap that like button and consider subscribing. It helps keep this corner of curiosity warm and welcoming. And please, let me know in the comments where you're listening from and what time it is in your part of the world. Are you settling in from a bustling city or finding quiet in a countryside evening? Fair warning, the universe of genetic editing is remarkably fascinating. I hope this isn't so interesting that it keeps you wide-eyed with wonder, but if it does, at least you'll drift off knowing something beautiful about the molecular poetry written in every cell of your body. So breathe deeply, let your thoughts settle like autumn leaves, and let's explore together what it means to become editors of life's ancient text. Close your eyes for a moment, if you're comfortable doing so, and imagine you're floating through the vast darkness of space. You're drifting past ancient stars and swirling galaxies, through clouds of cosmic dust that shimmer like powdered diamonds. But here's something remarkable, something that might make you pause in wonder as you drift through this celestial library. Everything you see out there in the cosmos, from the smallest grain of space dust to the largest spiral galaxy, follows the same fundamental laws of physics. Gravity pulls matter together. Energy flows from hot to cold. Light travels at exactly the same speed, whether it's streaming from a distant quasar or the gentle lamp beside your bed right now. But here on Earth, something even more extraordinary happened. About 4 billion years ago, in the warm, mineral-rich pools of our young planet, chemistry discovered how to write poetry. Life, you see, found a way to encode information using just 4 simple molecules. 4 chemical letters that, when strung together in endless combinations, could spell out the instructions for building a butterfly's wing, a redwood tree reaching toward the sky, or the intricate neural networks firing in your brain as you listen to these words. These 4 letters, adenine, thymine, guanine, and cytosine, are known to scientists simply as A, T, G, and C. But don't let their simplicity fool you. These aren't just chemicals floating in some distant laboratory. Right now, as you breathe slowly and settle into this moment, trillions upon trillions of these molecular letters are arranged in precise sequences inside every single cell of your body. Think about that for a moment. Every cell in your fingertips as they rest against your pillow, every cell in your heart as it beats its steady rhythm, every cell in your eyes as they perhaps grow heavy with sleep, each one contains the complete instruction manual for building you, written in this ancient 4-letter alphabet. It's as if every room in your house contained the complete architectural blueprints not just for that room, but for your entire home, your neighborhood, and indeed, the basic principles for building any structure that has ever existed or ever could exist. The wonder of it is almost too much to comprehend. Imagine if all of human literature, every poem ever written, every story ever told, every song ever sung could be encoded using just 4 symbols. Not the 26 letters of our alphabet, not the thousands of characters in Chinese writing, but just 4 simple marks. And yet, with these 4 letters, life has written the most complex and beautiful stories imaginable. The DNA in a single human cell, if you could stretch it out like a piece of thread, would extend about 6 feet. But it's coiled and folded so precisely that it fits inside a space smaller than the period at the end of this sentence. Picture this. If you could somehow print out all the genetic information in just one of your cells, using regular book-sized pages, you would have a library of about 1,000 volumes. Each cell, 1,000 books worth of information, written in 4 letters, compressed into a space so small you need a microscope to see it. But here's where the poetry truly lies. These aren't random arrangements of letters. They're instructions, passed down through an unbroken chain of life, stretching back billions of years. The same basic alphabet that spelled out the first simple bacteria still pulses through blue whales as they sing their haunting songs across ocean basins. the same four letters that guide a honeybee's dance also orchestrate the budding of cherry blossoms in spring. You carry within you the genetic echoes of every ancestor, every evolutionary step, every adaptation that allowed life to flourish and diversify across our planet. And all of it, written in this cosmic alphabet of just four letters. Sometimes, when I think about this, I imagine DNA as the universe's way of learning to write love letters to itself. Each species, each individual organism, is a unique poem written in this ancient language, variations on eternal themes of growth, survival, reproduction, and adaptation. Now, let's settle deeper into this idea and explore what it means to have such an instruction manual inside every cell. Imagine, if you will, that each of your cells is like a bustling kitchen in the world's most elaborate restaurant. But this isn't just any kitchen, it's a kitchen that has been perfecting its craft for billions of years. In this cellular kitchen, there sits a master cookbook. Not just any cookbook, but the most comprehensive collection of recipes ever assembled. This cookbook, your genome, contains somewhere between 20,000 and 25,000 different recipes. Each recipe is a gene, a specific sequence of those four letters we talked about, and each one contains instructions for making a particular protein. But here's where the metaphor becomes almost magical. Unlike the cookbooks in your own kitchen, which you might consult when you want to make Sunday dinner, this cellular cookbook is being read constantly, simultaneously, by multiple molecular chefs, all working at once. Some of these protein recipes are for the basic necessities of cellular life. There are recipes for the enzymes that break down the food you eat, transforming it into energy your cells can use. There are recipes for the structural proteins that give your cells their shape and strength, like the scaffolding that holds up a building under construction. Other recipes are more specialized, more exquisite in their precision. In the cells of your eyes, specific genes are being read to produce the proteins that capture light and transform it into the electrical signals your brain interprets as color and form. In your liver cells, entirely different recipes are being followed to produce the enzymes that detoxify harmful substances from your blood. And here's something beautiful to consider. As you rest there, the recipe being read most actively in your cells right now might be one for producing melatonin, that gentle hormone that helps ease you towards sleep. Your genome contains the instructions for this molecular lullaby, and your cells are faithfully following those ancient instructions, just as they have every night of your life. But there's something wonderfully mysterious about this cookbook. When scientists first began reading the human genome in the 1992nd and early 2002nd, they discovered something unexpected. The actual recipes, the genes that code for proteins, make up only about 2% of the entire book. 2%. The rest was initially called junk DNA, which now seems like calling the pauses between notes in a Beethoven symphony junk music. We've come to understand that much of this seemingly empty space contains the regulatory instructions, the directions, that tell the kitchen when to cook certain recipes, how much to make, and when to stop. It's like discovering that a cookbook contains not just the recipes themselves, but also detailed notes about when to prepare each dish, how to adjust for different seasons, what to cook when you're celebrating, what to make when you need comfort, and how to coordinate dozens of dishes so they all come together at exactly the right moment. Some of these regulatory regions are like dimmer switches, turning genes up or down depending on what the cell needs. Others are like timing circuits, ensuring that genes are activated in the right sequence as a cell grows and develops. Still others are like emergency switches, ready to activate stress response genes when the cell encounters danger. The beautiful inefficiency we once saw in the genome, all that seemingly wasted space, turned out to be a sophisticated control system, refined over billions of years of evolution. It's not empty space at all. It's the silence between the notes that makes the music meaningful. So here we are, resting in the glow of this understanding. We've explored the cosmic alphabet of life, written in just 4 chemical letters, but capable of infinite complexity. We've wandered through the cellular kitchens where genetic recipes are read and followed with exquisite precision. We've marveled at the copying miracle that preserves and passes on genetic information with almost unimaginable accuracy. For most of human history, this genetic text was written in a language we couldn't read, its messages encoded in molecules too small to see, its instructions carried out by processes we could only wonder about. But something remarkable has happened in just the last few decades. We've learned not only to read this ancient language, but to understand its grammar, its syntax, its subtle regulatory mechanisms. We've developed tools that can find specific sentences in this vast genetic library, tools that can highlight particular passages, tools that can even make careful edits to the text itself. But what happens when we want to become the editors of this ancient text? What happens when we develop the ability to reach into the very instruction manual of life and make deliberate changes? When we can correct the typos that cause disease, enhance the passages that promote health, or even write entirely new chapters in the story of what it means to be human? This is where our journey takes us next, into the realm of genetic editing, a technology so precise it can change a single letter in the 3 billion letter book of the human genome, so powerful it promises to heal diseases that have plagued humanity for millennia, and so profound in its implications that it asks us to reconsider what it means to be the authors of our own biological destiny. So let's continue our exploration together, diving deeper into this world where science fiction becomes science fact, where the stuff of dreams meets the reality of DNA, and where humanity is learning to write new verses in the ancient poem of life itself. The molecular scissors await, and with them, the possibility of healing, enhancement, and transformation beyond anything our ancestors could have imagined. But first, let us take a moment to appreciate the elegance of what already exists, this 4-letter alphabet that has written the story of life on Earth, including the story of you, listening here, now, in this quiet moment of discovery. In the next part of our journey, we'll meet the tools that make genetic editing possible, molecular machines borrowed from bacteria and refined by human ingenuity, capable of finding and changing specific sequences in the vast library of life. We'll discover how nature's own immune systems became our instruments of healing, and how the precision of these molecular tools is opening doors we never imagined could be opened. Rest easy now, knowing that the genetic text that makes you who you are has been preserved and protected by billions of years of evolutionary refinement. And when you're ready, we'll explore together what it means to become co-authors in the continuing story of life itself. Tonight, as you settle into whatever comfortable space holds you, perhaps a favorite chair where the light falls just so, or beneath covers that have learned the shape of your rest, I want to tell you a love story. But this isn't the kind of love story you might expect. This is a tale written in molecules and played out over billions of years. A story of conflict and adaptation that would eventually give humanity one of its most powerful tools for healing. It begins in a world almost unimaginably ancient, long before flowers bloomed or birds sang, even before the first complex cells learned to work together. Picture, if you will, the Earth as it was nearly 4 billion years ago, a planet of warm, mineral-rich seas and volcanic islands, where the first simple forms of life were just beginning to write their stories in that 4-letter alphabet we explored together. In this primordial world lived the bacteria, single-celled organisms that, despite their simplicity, were already accomplished chemists and engineers. They had learned to harvest energy from sunlight, to break down complex molecules for food, to reproduce with elegant efficiency. But in this ancient bacterial paradise, there lurked a constant threat: viruses. Now, you might think of viruses as we know them today, the ones that cause colds and flu, the microscopic invaders that our immune systems work so hard to defeat. But imagine viruses that are to bacteria what bacteria are to us. These bacteriophages, as scientists call them, are perhaps the most abundant life forms on Earth. They exist in numbers so vast that if you could somehow count all the bacteriophages in the ocean, you would find more of them than there are stars in the observable universe. And just as a master chef might have hundreds of variations on a basic recipe, your cells can read the same gene in different ways, producing slightly different proteins depending on what's needed at any given moment. Alternative splicing, it's called like having a recipe that can be modified on the fly to create dozens of different dishes from the same basic ingredients. Now, let me share with you one of the most extraordinary aspects of this genetic story. Something so remarkable that it might help you understand why scientists often speak of DNA with something approaching reverence. Every time one of your cells divides, which happens millions of times each day, as your body repairs and renews itself, that cell must make a complete copy of its entire genetic library. Not just a few pages, not just the recipes it uses most often, but every single letter of those 3 billion base pairs that make up the human genome. Imagine a monk in a medieval monastery, carefully transcribing an entire library by hand. Every word, every punctuation mark, every illustration must be copied with perfect precision. Now imagine that this monk must complete not just one book, but 3,000 books, and must finish the entire task in just a few hours. This is essentially what your cells accomplish every time they divide. In the warm, liquid environment of your body, molecular machines that are themselves made of proteins move along the DNA double helix, reading each letter and assembling a perfect complementary copy. The machinery that accomplishes this feat is breathtakingly elegant. DNA polymerase, the main copying enzyme, moves along the DNA strand like a tiny train on molecular tracks. It reads each letter and adds the matching partner A pairs with T, G pairs with C with astounding accuracy. But here's what might help you drift towards sleep with a sense of wonder. This process isn't happening somewhere far away in a laboratory. It's happening right now, inside your body, as you listen to these words. In your bone marrow, new blood cells are being born as stem cells copy their genetic instructions and divide. In your digestive tract, the cells that line your intestines are renewing themselves every few days. In your skin, new cells are constantly being created to replace the ones that are naturally shed. The fidelity of this copying process is extraordinary. DNA polymerase makes an error only about once in every 10 million letters it copies. But even that isn't precise enough for something as important as our genetic inheritance. So cells have evolved proofreading mechanisms, molecular editors that follow behind the copying machinery, checking for mistakes and correcting them. And yet, despite all these safeguards, occasional typos do slip through. A letter gets changed here, a short sequence gets deleted there, sometimes a section gets duplicated. These mutations, as we call them, are usually so rare and so small that they have no effect at all. But sometimes, very rarely, these tiny changes create something new. A protein that works slightly differently, a regulatory sequence that responds to a new signal, a subtle alteration that gives an organism some small advantage in surviving and reproducing. This is how evolution writes new chapters in the story of life, not through dramatic rewrites, but through tiny editorial changes accumulated over countless generations. The variations that make each person unique, that give you your particular eye color or the way your voice sounds when you laugh, all trace back to these gentle mutations that occurred in your ancestors and were preserved and passed down to you. Right now, as you rest there, your cells are engaged in this ancient dance of copying and dividing. The same molecular processes that preserve genetic information through countless generations of your ancestors are working to maintain the integrity of your own genetic code. There's something profoundly comforting about this, isn't there? You are connected to every living thing on Earth through this shared genetic alphabet, through these common molecular mechanisms that have been refined over billions of years. The DNA in your cells speaks the same fundamental language as the DNA in the trees outside your window, the bacteria in your gut, the birds that might be singing somewhere in the distance. You are part of an unbroken chain of life. Each link in that chain a successful copying of genetic information from one generation to the next. And tonight, as your body rests and repairs itself, this ancient copying machinery continues its work, ensuring that the unique combination of genes that makes you who you are will be preserved and maintained. Each bacteriophage is like a tiny spaceship, elegant in its geometric simplicity. It lands on a bacterial cell, injects its genetic material, and then commandeers the bacterial machinery to make copies of itself, often hundreds of copies, until the bacterial cell literally bursts from the pressure, releasing a new generation of viruses to continue their infectious cycle. For early bacteria, this was an existential crisis. How do you defend against an enemy that can reprogram your very cellular machinery? How do you survive when your attacker uses your own tools against you? And so, over millions of generations, through countless cycles of infection and death and rare survival, bacteria evolved something remarkable. They developed a memory system, a way to remember the genetic signatures of viruses that had attacked them before and to mount a rapid, precise defense when those same viruses returned. This bacterial memory system, refined over billions of years of evolutionary pressure, is what we now call CRISPR-clustered, regularly interspaced, short palindromic repeats. But don't let the technical name distract you from the beauty of what it represents. CRISPR is nature's own immune memory, a molecular filing system that bacteria use to keep records of their viral encounters. Picture it this way. When a bacterium survives a viral attack, it takes a small piece of the virus's genetic code, like keeping a wanted poster or a fingerprint, and files it away in a special section of its own DNA. This genetic archive grows over time, creating a molecular memory bank of all the viral enemies the bacterium or its ancestors have encountered. But memory alone isn't enough. The bacterium also needs a way to act on that memory, to recognize and destroy returning viral invaders. And so, alongside the memory system, bacteria evolved molecular scissors, proteins that could be guided to cut very specific DNA sequences with extraordinary precision. For billions of years, this system worked quietly in the microbial world, an elegant solution to an ancient problem. Bacteria passed their viral memories from generation to generation, each inheriting not just the basic tools for life, but also the accumulated wisdom of countless encounters with viral enemies. And then, in the early years of the 21st century, human scientists began to notice something extraordinary. They realized that this bacterial defense system, forged in the crucible of primordial warfare, could be adapted, modified, and repurposed as the most precise genetic editing tool ever discovered. There's something almost poetic about this, isn't there? The same molecular machinery that bacteria developed to defend themselves against viral invaders became humanity's instrument for healing genetic diseases. The tools of ancient warfare became instruments of modern medicine. Jennifer Doudna and Emmanuelle Charpentier, the scientists who would later win the Nobel Prize for their work in developing CRISPR as a research tool, often speak about the moment they realized the potential of what they had discovered. They were studying how bacteria defend themselves, trying to understand the fundamental mechanisms of this ancient immune system, when they recognized that these molecular scissors could be programmed to cut any DNA sequence they chose. It was as if they had discovered that a key crafted by bacteria to protect against one specific lock could be reshaped to open any door they wanted. The implications were staggering, not just for scientific research, but for treating genetic diseases, improving crops, and potentially reshaping life itself. The bacterial defense system that had evolved over billions of years in the struggle between microbes and viruses had become, in human hands, the foundation of a new era in biology. Ancient wisdom, preserved in the DNA of the smallest living things, had gifted humanity with the power to edit the very code of life. And so our love story, the billion-year dance between bacteria and viruses, becomes a story of human possibility. The molecular memory system that helped bacteria survive in a hostile world now helps us imagine a world where genetic diseases need not be permanent, where the typos in our genetic code can be carefully corrected, where the instructions for life itself can be edited with unprecedented precision. But to understand how this remarkable tool works, we need to meet its most important component, a protein that acts as both detective and surgeon, capable of finding one specific genetic sequence among billions and making exactly the cut we want it to make. Imagine for a moment that you're standing in the largest library ever built. This library contains not thousands or even millions of books, but 3 billion volumes, each one a single letter in the vast text of a human genome. The books stretch in every direction as far as you can see, arranged on shelves that reach impossibly high into the dim, quiet air. Somewhere in this vast collection is one specific sentence, perhaps just 20 letters long, that you need to find and edit. Maybe it's a sentence that, when corrected, could restore sight to the blind or strength to weakened muscles. Maybe it's a passage that, when rewritten, could prevent a devastating genetic disease from passing to the next generation. In a traditional library, this would be an impossible task. You might spend lifetimes searching through those 3 billion volumes, never finding the one sentence you seek. But in the molecular library of the living cell, there exists a librarian unlike any other, a protein called Cas9 that can navigate this vast collection with extraordinary precision and make exactly the editorial changes you specify. Cas9, the name comes from CRISPR-associated protein 9, is perhaps one of the most elegant molecular machines that evolution has ever produced. Picture it as a compact, roughly spherical protein about 5 nanometers across. To put that in perspective, if Cas9 were the size of a marble, a typical bacterial cell would be about as large as a football field. But size, in the molecular world, has little correlation with capability. Despite its tiny dimensions, CAS9 is a marvel of biological engineering, a protein that combines the functions of a search engine, a pair of scissors, and a word processor, all in one remarkably sophisticated package. The structure of CAS9 reveals the elegance of its function. Like a well-designed tool, every part serves a specific purpose. There are domains that bind to DNA, domains that interact with RNA guides, and most importantly, two distinct cutting domains that work together like the blades of molecular scissors. When CAS9 is activated and programmed with the right instructions, it begins a methodical search through the cellular library. It moves along the DNA double helix, testing each sequence it encounters against the specific pattern it has been programmed to recognize. Most of the time, the DNA sequence doesn't match, and CAS9 simply moves on, continuing its patient search. But when CAS9 encounters the exact sequence it's looking for, when it finds that one sentence among the 3 billion letters of the human genome, something remarkable happens. The protein undergoes a subtle but crucial change in shape, like a key fitting perfectly into a lock. This conformational change activates the cutting domains, and with extraordinary precision, CAS9 makes a clean, double-stranded cut in the DNA. Think of it as the most careful editor you could imagine, one who can find a specific typo in a library of books and correct it without disturbing a single other letter on the page. The precision is almost beyond comprehension. In a genome containing 3 billion base pairs, CAS9 can locate and cut at exactly the right spot, making an edit that affects only the specific sequence you want to change. But CAS9 doesn't work alone. Like any good librarian, it needs an address system, a way to know exactly where in the vast genetic library it should go to make its careful edit. This is where the true genius of the CRISPR system becomes apparent, because the targeting mechanism is as elegant as it is precise. The cutting ability of CAS9 is remarkable enough, but what makes the system truly revolutionary is its programmability. Unlike other molecular scissors that can cut only specific, predetermined sequences, CAS9 can be directed to virtually any location in the genome simply by providing it with the right guide. Scientists often describe CAS9 as having two main functional parts: a recognition system and a cutting system. The recognition system is what allows CAS9 to scan through DNA and identify its target sequence. But this recognition doesn't happen randomly or through some complex chemical affinity. Instead, CAS9 relies on a separate RNA molecule, a guide RNA, that contains the complementary sequence to whatever DNA target you want to hit. It's like giving a molecular bloodhound the scent of exactly what you want it to find. The guide RNA pairs with the target DNA through the same base pairing rules we discussed earlier, A with T, G with C, creating a short section of DNA-RNA hybrid that signals to CAS9, "This is the place. Make your cut here." When this recognition occurs, the cutting domains of CAS9 spring into action. One domain cuts one strand of the DNA double helix, while the other cuts the complementary strand, creating what scientists call a blunt-ended double-strand break. It's a clean cut, like using sharp scissors on paper rather than tearing it with your hands. The beauty of this system is that once the cut is made, the cell's own repair mechanisms take over. Cells have evolved sophisticated machinery for repairing broken DNA. They have to, given how often DNA damage occurs naturally. Sometimes this repair process simply glues the broken ends back together. Other times, if you provide a template, a corrected version of the sequence, the cell will incorporate the new information, effectively rewriting that section of its genetic code. This is how CAS9 became nature's word processor. It can delete harmful sequences, insert beneficial ones, or make precise letter-by-letter substitutions in the text of life. All of this happens at the molecular level, in the warm, crowded environment of the living cell, with a precision that would be remarkable even in the most controlled laboratory setting. And here's something that might help you appreciate the true elegance of this system. CAS9 evolved not to edit genomes for human benefit, but as part of that ancient bacterial immune system we discussed. The same protein that bacteria use to cut up invading viral DNA is the one that scientists have repurposed to correct genetic defects, enhance crop resistance, and potentially treat thousands of inherited diseases. Evolution, working over billions of years, refined CAS9 to be exactly what we needed it to be, even though evolution, of course, had no knowledge of our future needs. It's as if the history of life on Earth has been preparing all along to give us the tools we would need to become gentle editors of our own genetic destiny. But to truly understand how this molecular word processor operates, we need to explore its guidance system, the elegant mechanism that tells CAs9 exactly where to go in the vast library of our genes. There's something almost magical about the way information finds its target in the molecular world of the cell. Right now, as you rest there, countless molecular messages are being delivered with extraordinary precision throughout your body. Hormones released in one organ travel through your bloodstream to affect specific cells hundreds of miles away in biological terms. Nerve signals race along precisely defined pathways to coordinate the rhythm of your breathing, the beating of your heart, the complex dance of thoughts forming in your mind. But perhaps nowhere is this precision more beautiful than in the guidance system that directs CAs9 to its target, a short RNA molecule that acts like a molecular GPS, leading our protein scissors to exactly the right address in the vast city of our genome. This guide RNA, scientists call it gRNA, is typically only about 20 nucleotides long, 20 letters in that same four-letter alphabet we've been exploring together. In the context of a genome containing 3 billion base pairs, 20 letters might seem almost insignificantly small, but those 20 letters contain enough information to specify a unique address in the entire human genome. Think about that for a moment. With just 20 letters, chosen carefully from an alphabet of 4, we can create an address so specific that it appears nowhere else in the 3 billion letters of human DNA. It's like having a postal code so precise that it identifies not just a city or a street, but one specific room in one specific house among all the buildings on Earth. The elegance of this addressing system becomes clear when you understand how guide RNA does its work. The RNA molecule is designed to be complementary to the target DNA sequence. Every A in the guide RNA pairs with a T in the target DNA, every G pairs with a C. This isn't random pairing, it's the same molecular recognition system that allows DNA to replicate itself with such fidelity. Picture the guide RNA as a molecular key, shaped to fit exactly one lock in the entire genome. As CAs9 moves through the cellular library, the guide RNA it carries tests each potential binding site, like trying a key in door after door. Most of the time, the molecular shapes don't match, the key doesn't fit the lock, and CAs9 continues its search. But when the guide RNA encounters its perfect complement in the target DNA, something beautiful happens. The two molecules recognize each other with the specificity of old friends. The RNA and DNA strands align, base by base, forming a short section of RNA-DNA hybrid that is as stable as it is precise. This moment of recognition when the guide RNA finds its complementary sequence in the target DNA is like watching puzzle pieces click into place. The molecular shapes fit together so perfectly that they exclude all other possibilities. There's no ambiguity, no uncertainty. This is the target. This is where the edit should be made. But the guide RNA does more than just identify the target location. It also undergoes a subtle change in shape when it binds to its complementary DNA. And this change signals to CAs9 that the correct target has been found. It's like a molecular handshake between the guide RNA and the target DNA, with CAs9 sensing this interaction and responding by activating its cutting domains. The specificity of this system is almost beyond belief. A guide RNA designed to target one specific gene sequence will ignore the millions of other sequences in the genome, even sequences that differ by only a single letter. This selectivity is what makes CRISPR editing possible without it. CAs9 would make cuts randomly throughout the genome, causing chaos rather than precise editing. Scientists have spent years studying and refining this guidance system, learning how to design guide RNAs that maximize accuracy while minimizing off-target effects. They've discovered that the position of mismatches matters. Some locations in the guide RNA are more tolerant of differences than others. They've learned that the local chromatin structure around the target site can affect accessibility, and that the specific sequence context can influence cutting efficiency. But perhaps the most remarkable thing about guide RNA is how it democratizes genetic editing. Before CRISPR, changing specific DNA sequences required complex, custom-designed proteins that might take months or years to engineer. With CRISPR, Targeting a new location requires only the synthesis of a new guide RNA, a process that can be completed in days or even hours. This simplicity has transformed genetic editing from an art practiced by a few specialists into a tool accessible to researchers around the world. Today, in laboratories across the globe, scientists are designing guide RNAs to target the genetic causes of blindness, the mutations that lead to sickle cell disease, the DNA sequences that make cancer cells resistant to treatment. Right now, as we speak, molecular choreography is taking place in research labs and clinical trials around the world. Guide RNAs are finding their targets with extraordinary precision, directing CAs9 to make careful edits in the genomes of cells that might someday help heal diseases that have plagued humanity for millennia. Each successful targeting event, each moment when a guide RNA finds its complement and directs CAs9 to make its precise cut, represents the culmination of billions of years of evolution and decades of human ingenuity. The same base pairing rules that allow DNA to replicate faithfully, generation after generation, now allow us to edit genetic information with unprecedented control. But the story of molecular scissors doesn't end with CAs9 and guide RNA, remarkable as they are. Just as evolution continues to refine and improve biological systems over time, human scientists have been working to develop even more precise, even more versatile tools for genetic editing. The original CRISPR system, borrowed from bacteria and refined in human laboratories, was just the beginning. What came next would make genetic editing even more precise, even more gentle, even more like the careful work of a master editor polishing a treasured manuscript. There's something deeply human about the way we take a powerful tool and immediately begin imagining how to make it better. Give us fire, and we'll learn to forge metal. Give us the wheel, and we'll dream of locomotives and automobiles. Give us molecular scissors that can cut DNA with extraordinary precision, and we'll start wondering, what if we could edit genetic text without cutting it at all? This is exactly what happened as scientists around the world began working with CRISPR-Cas9. The system was revolutionary, yes, capable of making precise cuts in DNA and allowing targeted edits that had been impossible just years before. But as researchers used these molecular scissors in their laboratories, they began to notice something. Sometimes, making a cut isn't the most elegant solution to a genetic problem. There are scientists working on protein editors that can modify the enzymes and structural molecules in our cells without changing the genes that encode them. There are researchers developing epigenetic editors that can change how genes are regulated without altering the underlying DNA sequence at all. Each of these tools represents a different approach to the fundamental challenge of genetic editing. How do we make precise, beneficial changes to the incredibly complex systems that govern life? The molecular scissors we've explored together, from the original bacterial CRISPR systems to the sophisticated base and prime editors developed in human laboratories, represent humanity's growing ability to read, understand, and gently modify the text of life itself. We've learned to borrow tools from ancient bacterial immune systems and refine them into instruments of healing. We've developed ways to target any sequence in the human genome with extraordinary precision. We've created molecular editors that can make changes as gentle as correcting a single misspelled letter, or as comprehensive as rewriting entire paragraphs in the Book of Life. Now that we understand the tools, the molecular scissors, the guidance systems, the increasingly sophisticated editors that scientists have developed, it's time to explore what happens when we use them. How do these tools transform from laboratory curiosities into treatments for real diseases affecting real people? What does it mean to take the bacterial defense systems refined over billions of years and use them to restore sight to the blind, to give strength to those weakened by genetic disease, to offer hope to families facing devastating inherited conditions? The tools are elegant, powerful, and increasingly precise. But tools, no matter how sophisticated, are only as meaningful as the purposes to which we put them. In our next exploration together, we'll discover how these molecular scissors are being used to heal, to restore, and to rewrite some of the most heartbreaking chapters in the long story of human suffering. The science is remarkable, but the human stories, the patients who volunteer for the first trials, the families who dare to hope, the children who might grow up free from genetic diseases that have plagued their bloodlines for generations, these stories are where the true power of genetic editing becomes clear. So let us continue our journey together, from the elegant mechanics of molecular editing to to the profound human implications of our growing power to rewrite the code of life itself. The tools are ready. The precision is unprecedented, and the possibilities for healing, for hope, for human flourishing, stretch out before us like an open book, waiting to be written. There's a particular quality to hope that emerges in the quiet hours of the night, when the world has settled into stillness and our thoughts turn to the deepest questions of what it means to be human. Tonight, as you rest in whatever space holds you gently, I want to share with you some of the most profound stories in modern medicine stories of how the molecular tools we've explored together are being used not just to edit genes, but to rewrite the narratives of human suffering. These are stories where the abstract beauty of molecular scissors meets the very real experiences of patients and families who have lived with genetic diseases that, until recently, seemed as permanent as the stars. They are stories of children who couldn't see learning to perceive light, of blood cells learning to carry oxygen properly, of immune systems being taught to recognize and destroy cancer. But perhaps most remarkably, these are stories that illustrate how the smallest changes a single letter edited in the vast library of our genetic code can transform entire lives. They remind us that in the intricate dance of molecules that makes us who we are, even the tiniest misstep can echo through every aspect of our existence, and even the most precise correction can restore possibilities that seemed forever lost. Let me tell you about a single letter in the human alphabet of life, one letter among the 3 billion that spell out our genetic instructions, and how changing that letter has begun to rewrite the story of one of the most devastating genetic diseases known to medicine. The letter is an A that should be a T, sitting in position 20 of the beta-globin gene on chromosome 11. Just one letter, in one gene, among the 25,000 genes that make up the human genome. But this single substitution, this tiny typo in the book of life, causes a cascade of molecular consequences that can fill an entire lifetime with pain. Consider, for example, a genetic disease caused by a single letter change in the DNA, one A that should be a G or one C that should be a T. Using traditional CRISPR-Cas9 to fix such a problem would be like using a sledgehammer to adjust a picture frame. You could make the cut, provide a corrected template, and hope the cell's repair machinery would incorporate the fix correctly. But cellular repair can be unpredictable, sometimes making unintended changes or failing to incorporate the correction at all. What if, scientists wondered, we could develop tools that could change single letters in the genetic code directly, without making any cuts at all? What if we could perform genetic editing with the precision of a pencil eraser rather than molecular scissors? This question led to the development of what scientists call base editors, modified versions of Cas9 that can change single DNA letters with extraordinary precision. Instead of cutting the DNA double helix, these base editors chemically convert one base to another right in place, like a molecular spell checker that can fix typos without disturbing the surrounding text. The first base editors, developed by David Liu's laboratory at the Broad Institute, could perform cytosine to thymine edits, essentially changing Cs to Ts anywhere in the genome that a guide RNA could direct them. Later versions could perform adenine to guanine edits, expanding the toolkit to cover the most common types of disease-causing mutations. Picture the elegance of this approach. A base editor, guided by its RNA address system, lands at exactly the right location in the genome. But instead of cutting, it gently changes one chemical letter to another, making its edit and moving on, leaving the DNA intact except for that single, precise modification. These base editors work by fusing Cas9, or related proteins, with enzymes that can perform specific chemical conversions. The Cas9 component provides the targeting. It still uses guide RNA to find the right location, but its cutting function has been disabled. Instead, attached to the Cas9 is an enzyme that can chemically modify DNA bases, changing their identity without breaking the DNA backbone. But even base editors weren't the end of the story. As scientists worked with these tools, they realized that some genetic corrections required more than just single-letter changes. Sometimes you need to insert new DNA sequences, delete small sections, or make more complex edits that couldn't be accomplished with simple base conversion. This led to the development of prime editors tools that represent perhaps the closest thing we have to a true molecular word processor. Prime editors can make small insertions, precise deletions, and any combination of base changes within a small region of the genome, all without making double-strand breaks in the DNA. The way prime editing works is almost like molecular copy and paste. The system includes a modified CA's protein attached to a reverse transcriptase enzyme, the same type of enzyme that some viruses use to copy their RNA genomes into DNA. The guide RNA for prime editing is special too. It contains not just the addressing information, but also a template for the desired edit. When the prime editor finds its target, it makes a small nick in one strand of the DNA and uses its reverse transcriptase to copy the editing template into the genome. The result is a precise edit that can involve multiple changes within a small region, insertions, deletions, and substitutions, all accomplished in a single, coordinated operation. What's remarkable about these newer editing tools is how they expand our ability to correct genetic problems with increasing gentleness and precision. We've moved from molecular sledgehammers to molecular scalpels, to molecular pencils to molecular word processors, each new tool allowing more refined and less disruptive edits to the code of life. And the pace of innovation continues to accelerate. Even as I share these words with you, scientists in laboratories around the world are developing new variants of these tools, base editors that can perform new types of chemical conversions, prime editors with improved efficiency and precision, and entirely new approaches to genetic editing that we're only beginning to imagine. Some researchers are working on tools that can edit RNA instead of DNA, allowing temporary modifications that don't permanently alter the genome. Others are developing systems that can edit multiple locations simultaneously, or that can be controlled with light or small molecules, turning genetic edits on and off at will. When this letter is wrong, the red blood cells that should carry oxygen smoothly through our circulation instead become twisted into the shape of crescents or sickles. These deformed cells are rigid where they should be flexible, sticky where they should flow freely. They clump together in tiny blood vessels, blocking the flow of oxygen-rich blood to tissues throughout the body. The result is sickle cell anemia, a disease that affects millions of people worldwide, primarily those of African descent, whose ancestors developed this genetic change as partial protection against malaria. It's one of evolution's cruel ironies. A mutation that provided some survival advantage in malarial regions became, in its full form, a source of lifelong suffering. People with sickle cell disease experience episodes of excruciating pain as their misshapen blood cells block circulation. They suffer from organ damage, strokes, and infections. Many don't live to see their 50th birthday. And until very recently, there was little medicine could do except manage the symptoms and hope for the best. But in 2019, a woman named Victoria Gray became one of the first people in the world to receive a CRISPR-based treatment for sickle cell disease. Her story, still unfolding as I share these words with you, represents a turning point not just in her own life, but in our understanding of what genetic medicine can accomplish. Victoria had lived with sickle cell disease for decades. She had endured countless pain crises, hospitalizations, and the daily uncertainty of living with a genetic condition that could flare without warning. She had watched the disease limit her possibilities, affect her ability to work, and cast a shadow over her family's future. The treatment Victoria received was elegantly simple in concept, though extraordinarily complex in execution. Doctors removed some of her bone marrow cells, the stem cells that give rise to all the blood cells in her body. In the laboratory, these cells were treated with CRISPR tools designed to make a very specific edit. But here's where the story becomes particularly beautiful. Rather than trying to directly fix that single wrong letter in the beta-globin gene, scientists took a different approach. They used CRISPR to reactivate a different gene, one that produces fetal hemoglobin, the type of oxygen-carrying protein we make before birth. You see, all of us carried perfect hemoglobin when we were developing in the womb. Fetal hemoglobin doesn't sickle, doesn't cause pain crises, doesn't lead to organ damage. But normally, our bodies switch off the production of fetal hemoglobin shortly after birth, replacing it with adult hemoglobin. By using CRISPR to remove the genetic switch that turns off fetal hemoglobin production, scientists could restore Victoria's blood cells' ability to make the healthy oxygen carriers she had possessed before birth. It was like finding a backup copy of a crucial software program and reactivating it after the main version had become corrupted. The edited cells were then infused back into Victoria's body, where they took up residence in her bone marrow and began producing new, healthy blood cells. Month by month, her blood began to change. The percentage of normal, flexible red blood cells increased. The painful sickle-shaped cells became rarer and rarer. As I share this story with you, Victoria Gray has been free from pain crises for several years now. Her blood carries oxygen efficiently throughout her body. She has been able to return to work, to plan for the future, to imagine possibilities that sickle cell disease had once made impossible. There's something almost mystical about the idea that a single letter change, one edit among 3 billion possibilities, could so completely transform a life. It reminds us that we exist in a delicate balance of molecular interactions, where the smallest perturbation can ripple through our entire biological symphony. But it also speaks to the extraordinary precision of the tools we've developed. In Victoria's case, scientists weren't randomly editing her genome, hoping for the best. They were making a specific, targeted change based on decades of research into the molecular basis of sickle cell disease and the normal development of our oxygen-carrying systems. Victoria's story is still being written, and scientists are carefully monitoring not just her health, but that of other patients who have received similar treatments. Each success builds our confidence. Each challenge teaches us how to make the next intervention even safer and more effective. But sickle cell disease is just one example of how genetic editing is beginning to heal conditions that were once considered permanent features of human existence. Let me share with you another story, one that literally brings light into darkness. There are moments in medicine that transcend the technical details of treatments and touch something fundamental about what it means to be human. One of these moments occurs when someone sees clearly for the first time in their life, when genetic blindness, passed down through generations like a family curse, is lifted by the careful application of molecular editing tools. Leber congenital amaurosis, let's call it LCA, because the name matters less than the story it represents, is a group of inherited conditions that rob children of sight from birth or early infancy. These aren't diseases caused by injury or infection, but by mutations in genes that are essential for the complex molecular machinery of vision. Picture, for a moment, the extraordinary process that allows you to see. Light enters your eye and strikes specialized cells in your retina called photoreceptors. These cells contain proteins that literally change shape when light hits them, triggering a cascade of electrical signals that your brain interprets as images, colors, movement, depth. The genes mutated in LCA encode various components of this visual machinery. Some code for proteins that help photoreceptor cells maintain their structure. Others produce enzymes involved in the chemical cycles that reset our vision after each flash of light detection. Still, others guide the development of the retina itself during embryonic growth. When any of these genes carries a mutation that disrupts its function, the result is profound visual impairment. Children born with LCA typically have very little functional vision. They may be able to detect bright light or perceive vague shapes, but they cannot see faces, cannot read, cannot navigate the visual world that most of us take for granted. But in recent years, clinical trials have begun using genetic editing approaches to restore vision to children and adults with certain forms of LCA. The results have been nothing short of miraculous, though that word, overused as it often is, seems almost inadequate to describe the experience of giving sight to someone who has never seen. One of the most promising approaches involves editing genes directly in the retina. Scientists have developed ways to deliver CRISPR tools to the photoreceptor cells of the eye, where they can correct mutations that prevent normal vision from developing. In some early trials, researchers have used base editors, those gentle molecular pencils we discussed, to correct single-letter changes in genes like CP290, which is mutated in about 15% of LCA cases. The treatment involves a delicate injection directly into the eye, delivering the genetic editing tools to the cells that need them most. The first signs of improvement often come weeks or months after treatment, as the edited cells begin producing functional proteins and the visual system slowly awakens. Patients report being able to detect light sources they couldn't see before. They begin to make out shapes and movements that had been invisible to them. But perhaps the most profound changes are harder to measure in clinical trials. There's the moment when a child sees their parent's face clearly for the first time. The experience of watching a sunset, seeing the interplay of colors across the sky, understanding why people write poetry about the beauty of the natural world. Imagine, if you can, what it might be like to see color for the first time. Not to understand it as a concept, but to actually experience the visual sensation of red or blue or green, to discover that the world is painted in hues you never knew existed, that every object around you carries information encoded in wavelengths of light you had never been able to decode. There are children participating in these clinical trials who are learning that grass is green, that the sky is blue, that their mother's eyes are brown or hazel or grey. They're discovering that facial expressions carry meaning visible in the curve of a smile or the crinkle around someone's eyes when they laugh. The clinical results, measured in terms of light sensitivity and visual acuity, tell only part of the story. The real impact is in the transformation of daily life, the ability to navigate familiar spaces with confidence, to read expressions on loved ones' faces, to participate in visual experiences that connect us to our communities and our culture. Of course, not every patient experiences dramatic improvement, and the treatments are still experimental, still being refined. Vision is extraordinarily complex, and genetic blindness can result from dozens of different mutations in dozens of different genes. Each case requires careful analysis to determine whether genetic editing might help and which specific approach might be most effective. But the early results give us glimpses of a future where genetic blindness need not be permanent, where children born with LCA might grow up with functional vision, where families need not pass these conditions from generation to generation. The restoration of vision through genetic editing represents hope not just for those directly affected, but for all of us who recognize the profound importance of our senses in how we experience and understand the world. It's a reminder that genetic medicine isn't just about fixing broken genes, it's about restoring human capabilities, expanding possibilities, and giving people the tools they need to live fuller lives. But genetic editing isn't limited to correcting inherited diseases. Some of the most dramatic applications involve training our own immune systems to become more effective healers and protectors. Your immune system is, in many ways, the most sophisticated army ever assembled. It patrols your body constantly, identifying and eliminating threats that could harm you. It remembers previous encounters with dangerous microorganisms and mounts faster, more effective responses when those threats return. It can distinguish between your own healthy cells and foreign invaders with extraordinary precision. But cancer presents a unique challenge to this remarkable defense system. Cancer cells aren't foreign invaders from outside your body, they're your own cells that have gone rogue, accumulating mutations that allow them to grow uncontrollably and spread to new locations. Because cancer cells originate from normal cells, they often look very similar to healthy tissue. It's as if criminals were wearing perfect disguises, allowing them to move through your body undetected by the immune system that should be protecting you. Your T cells, the specialized immune cells that normally identify and destroy threats, often fail to recognize cancer cells as dangerous. But what if we could remove those disguises? What if we could give your immune system a wanted poster with the cancer's face clearly displayed, along with instructions to hunt down and eliminate every cell that matches that description? This is essentially what CAR T-cell therapy accomplishes, and it represents one of the most dramatic applications of genetic editing in medicine today. CAR T therapy begins by collecting T cells from a cancer patient's blood. These are the same immune cells that should be fighting the cancer but haven't been able to recognize it effectively. In the laboratory, scientists use genetic editing tools to modify these T cells, giving them new capabilities. The modification involves inserting genes that produce what's called a chimeric antigen receptor, CAR, on the surface of the T cells. Think of this receptor as a sophisticated molecular antenna that can detect specific proteins found on cancer cells but not on normal, healthy cells. Once the T cells have been equipped with these new receptors, they're grown in large numbers in the laboratory and then infused back into the patient. Now, these enhanced immune cells have a clear target. They can recognize cancer cells with precision and destroy them with the full force of the immune system. The results in some patients have been nothing short of extraordinary. Children with acute lymphoblastic leukemia who had failed multiple rounds of conventional treatment have achieved complete remissions after CAR T therapy. Adults with aggressive lymphomas that seemed certain to be fatal have seen their cancers disappear entirely. Let me tell you about Emily Whitehead, who became the first pediatric patient to receive CAR T-cell therapy in 2012. Emily was just 6 years old when she was diagnosed with acute lymphoblastic leukemia. She had already undergone months of chemotherapy, but the leukemia kept returning, more aggressive each time. When Emily received her genetically modified T cells, the initial response was dramatic and frightening. Her immune system, now equipped with powerful cancer-fighting capabilities, launched such an intense attack against her leukemia that she became critically ill from the inflammatory response. But when the storm passed, something remarkable had happened. The leukemia was gone, completely gone. Emily's own immune system, enhanced by genetic editing, had eliminated every detectable cancer cell in her body. Emily is now a teenager, more than a decade cancer-free. She plays sports, goes to school, dreams about her future, all possibilities that seemed lost when her leukemia appeared resistant to conventional treatment. The T cells that were edited to fight her cancer are still circulating in her body, standing guard against any return of the disease. Of course, California RT therapy doesn't work for everyone, and it's not without risks. The same immune activation that can eliminate cancer can also cause serious side effects. Some patients experience severe inflammatory reactions, while others develop complications from the depletion of normal, healthy B cells, along with the cancerous ones. And CAR T therapy is currently limited to certain types of blood cancers, leukemias, and lymphomas, where the target proteins are well defined and accessible to modified immune cells. Solid tumors like lung cancer, breast cancer, and brain tumors present additional challenges that scientists are still working to overcome. But the principle has been established. We can edit a patient's own immune cells to make them more effective cancer fighters. We can train the body's natural defenses to recognize and eliminate threats they previously couldn't detect. This approach represents a fundamental shift in how we think about cancer treatment. Instead of using external therapies like chemotherapy or radiation to attack cancer cells, we're enhancing the patient's own immune system to do the job more effectively. Scientists are now working on CAR T therapies for solid tumors, developing new targeting strategies, and exploring ways to make the treatments safer and more widely applicable. Each success teaches us more about how to harness the power of genetic editing to transform cancer from a death sentence into a manageable condition. The story of CAR T therapy reminds us that genetic editing isn't just about correcting inherited diseases. It's also about enhancing our natural biological capabilities, giving our own cells new tools to protect and heal our bodies. But some of the most poignant applications of genetic editing involve conditions where the missing piece isn't a single gene or protein, but an entire biological capability that has been lost to genetic mutation. There are few things more fundamental to human experience than the ability to move our bodies with intention and grace. From the first tentative steps of a toddler to the elegant movements of a dancer, from the simple act of reaching for a book to the complex coordination required to ride a bicycle, movement defines much of how we interact with the world around us. For families affected by Duchenne muscular dystrophy, this fundamental capability is gradually stolen away by a single missing protein. Duchenne muscular dystrophy, let's call it DMD, is caused by mutations in one of the largest genes in the human genome, a gene that produces a protein called dystrophin. This protein acts like molecular scaffolding, helping to maintain the structural integrity of muscle fibers during the constant cycles of contraction and relaxation that allow movement. When dystrophin is missing or non-functional, muscle fibers become fragile. Each contraction causes tiny tears in the muscle membrane. Over time, these tears accumulate, leading to inflammation, scarring, and the progressive replacement of functional muscle tissue with fibrous scar tissue. Boys with DMD typically develop normally in their early years. They learn to walk, to run, to play like any other child. But usually by age 3 or 4, parents begin to notice subtle signs. Their child may fall more often, have difficulty climbing stairs, or struggle to keep up with playmates. The progression is heartbreakingly predictable. By age 10 or 12, most boys with DMD need wheelchairs for mobility. The weakness spreads from the legs to the arms, affecting the ability to perform daily tasks independently. Eventually, the muscles that control breathing and heart function are affected as well. For decades, families facing DMD had few options beyond supportive care and the hope that their children would have as much time and quality of life as possible. But genetic editing is beginning to change this narrative. offering the possibility of restoring muscle function by correcting the underlying genetic defect. One of the most promising strategies involves using CRISPR tools to perform what scientists call exon skipping. The dystrophin gene is enormous 79 exons that must be properly joined together to produce functional protein. Many DMD mutations disrupt this joining process, preventing the production of any functional dystrophin at all. But here's where genetic editing offers an elegant solution. By using CRISPR to remove specific exons that contain or surround the mutation, scientists can often restore the gene's ability to produce a shortened but functional version of dystrophin. Think of it like editing a sentence that has become unreadable due to a few corrupted words. Instead of trying to fix each corrupted word individually, editors remove entire phrases, creating a shorter sentence that still conveys the essential meaning. In early clinical trials, researchers have used this approach to restore dystrophin production in muscle cells from DMD patients. The edited cells begin producing the protein that has been missing throughout these patients' lives, potentially halting or even reversing the progressive muscle weakness that defines the condition. Imagine what this might mean for a family that has watched their child's mobility decline year by year. The possibility that genetic editing could restore muscle function, that a boy who had lost the ability to walk might regain his independence, that the progression of weakness might be halted before it affects vital functions like breathing and heart rhythm. Of course, the reality is more complex than our hopes might suggest. DMD affects muscles throughout the body, and delivering genetic editing tools to all affected tissues remains a significant challenge. The heart muscle, the diaphragm, the muscles that control posture and fine motor skills, all must be reached for treatment to be fully effective. Early clinical trials are showing promising results, but they're also teaching us about the complexities of treating a systemic muscle disorder. Some patients show measurable increases in dystrophin production and improvements in muscle function. Others respond less dramatically, reminding us that genetic editing, while powerful, is not yet a perfect solution for all genetic diseases. But the progress is real, and it's accelerating. Researchers are developing new delivery methods to get genetic editing tools to muscles throughout the body. They're refining their techniques to make larger corrections to the dystrophin gene. They're exploring combination approaches that might address not just the missing protein, but also the inflammation and scarring that accumulate over years of muscle damage. For families affected by DMD, each advance in genetic editing research represents not just scientific progress, but renewed hope for their children's futures. Clinical trials that might restore muscle function, treatments that could halt disease progression, therapies that might allow children to grow up with capabilities their parents had thought were lost forever. The story of DMD research illustrates something important about genetic medicine. Progress often comes in increments rather than sudden breakthroughs. Each study teaches us more about how to deliver treatments effectively, how to optimize the genetic edits we make, how to support patients through the complex process of cellular repair and regeneration. But it also reminds us of the profound courage required of families who participate in clinical trials, who allow their children to be among the first to try experimental treatments, who balance hope with the uncertainty that comes with any pioneering medical intervention. Before we continue our exploration together, I want to take a moment to acknowledge something that often gets lost in the technical details of genetic editing and the excitement of scientific progress. Behind every story I've shared with you tonight, behind every clinical trial, every breakthrough, every carefully documented improvement in a patient's condition are human beings making extraordinarily courageous choices. Think about Victoria Gray, living with decades of sickle cell pain, deciding to become one of the first people in the world to have her genes edited with CRISPR technology. Consider the parents of children with LCA, enrolling their sons and daughters in experimental trials that might restore vision or might not work at all. Reflect on the families affected by muscular dystrophy, watching their children's strength fade year by year, choosing to participate in genetic editing studies because hope, however uncertain, feels better than resignation. These patients and families are pioneers in the truest sense. They're not just receiving experimental treatments, they're helping to establish whether genetic editing can safely and effectively treat human disease. Every piece of data from their experience will inform future treatments, potentially helping thousands of other patients who face similar diagnoses. And then there are the researchers, the scientists who spend years developing and refining genetic editing tools, often working with conditions so rare that each patient represents a significant portion of the entire population affected by that disease. These researchers must balance scientific rigor with compassion, careful methodology with urgent hope. There are the physicians who conduct clinical trials, who must explain complex experimental procedures to anxious families, who monitor patients carefully for both beneficial effects and potential complications. They carry the weight of responsibility that comes with offering cutting-edge treatments while honestly acknowledging the uncertainties involved. There are the laboratory technicians who culture cells, prepare genetic editing reagents, and perform the countless quality control tests that ensure experimental treatments meet the highest safety standards. There are the regulatory scientists who review trial protocols, ensuring that patient safety remains the highest priority even as we push the boundaries of what's medically possible. And there are the families who don't qualify for current trials, who watch the progress of genetic editing research with hope for future treatments that might help their own loved ones. There are the advocacy organizations that fund research, educate families, and work to ensure that genetic editing advances translate into accessible treatments for all who need them. Each of these groups, patients, families, researchers, clinicians, advocates, plays an essential role in the careful, methodical process of transforming genetic editing from a laboratory technique into a medical reality. Their collective courage, patience, and dedication make possible the stories of healing we've explored together. It's also important to acknowledge that not every story in genetic medicine has a happy ending. Clinical trials sometimes fail. Treatments that work in the laboratory don't always translate to effective therapies in patients. Some patients experience side effects that outweigh the benefits of treatment. But even the trials that don't succeed as hoped contribute essential knowledge to our understanding of genetic editing. They teach us about the complexities of human biology, the challenges of delivering treatments to specific tissues, the importance of careful patient selection and monitoring. There's something profoundly human about the willingness to try, to hope, to participate in the slow but steady advance of medical knowledge, even when success isn't guaranteed. It reflects our deepest values, our commitment to healing, our refusal to accept suffering as inevitable, our belief that human ingenuity can solve even the most challenging problems. As we've seen in the stories of sickle cell disease, inherited blindness, cancer, and muscular dystrophy, genetic editing is beginning to transform conditions that were once considered permanent features of human existence. Single letters changed in the vast library of our genetic code are restoring sight, eliminating sickle-shaped blood cells, training immune systems to fight cancer, and potentially giving strength back to weakened muscles. But these applications of genetic editing remarkable as they represent just the beginning of what may be possible. As our tools become more precise, our understanding deeper, and our delivery methods more sophisticated, the scope of genetic medicine will continue to expand. The courage of today's patients and the dedication of today's researchers are laying the foundation for a future where genetic diseases need not define entire lives, where the accidents of inheritance can be corrected with precision and compassion, where the molecular tools we've borrowed from ancient bacteria and refined in human laboratories can be used to heal suffering that has persisted for generations. In our next exploration together, we'll delve into some of the profound questions that arise as genetic editing becomes more powerful and more widely available. How do we ensure that these treatments benefit everyone who needs them? What happens when we can edit not just disease-causing mutations, but genetic variants that affect more complex traits? How do we navigate the ethical landscape of technology that gives us unprecedented power over our own biology? For now, let us rest in gratitude for the courage of those who go first, who volunteer for experimental treatments and clinical trials, who allow their experiences to light the way for others following behind them. Their stories remind us that medical progress isn't just about molecular tools and laboratory techniques, it's about human beings choosing hope, embracing uncertainty, and working together to write new chapters in the ancient story of healing. The genetic editing revolution is built on foundations of courage, compassion, and collaboration. And perhaps that's the most important lesson of all. There are moments in the quiet hours before dawn when our thoughts turn to the deepest questions of human existence, questions that have no easy answers. that invite us to explore the spaces between what we can do and what we should do. Tonight, as you rest in whatever peaceful space holds you, we enter such a space together, a place where the remarkable power of genetic editing meets the timeless complexities of human wisdom and moral responsibility. We've explored the elegant mechanisms of molecular scissors and witnessed their power to heal conditions that have caused suffering for generations. But with great power comes profound responsibility, and genetic editing raises questions that humanity has never had to answer before. Tonight, we'll walk together through what I think of as an ethical labyrinth, a complex maze of moral considerations, where every path forward opens onto new questions, new perspectives, new ways of understanding our responsibility to future generations. Imagine two different kinds of letters you might write. The first is a personal note to a friend, expressing your thoughts in this moment. When you've written it and sent it, the words exist for a time, touching one life, and then eventually fade into memory. The second kind of letter is different entirely. Imagine writing something that would be copied exactly and passed down through your family line for generations, every word preserved, every comma passed along unchanged, becoming part of your family's permanent record. In genetic editing, we face a similar choice. Most of the healing applications we've explored treating sickle cell disease, restoring vision, enhancing immune cells to fight cancer, involve somatic cell editing. These changes affect only the patient receiving treatment. They don't get passed on to future generations. But there's another possibility: germline editing. This involves making changes to eggs, sperm, or early embryos, changes that would be incorporated into every cell of the resulting person's body, including the cells that would eventually give rise to their own children. This distinction became painfully clear in late 2018 when Chinese scientist He Jiankui announced that he had used CRISPR to edit the genes of human embryos that were then brought to term, resulting in the birth of twin girls whose genetic code had been deliberately altered. He Jiankui was not a reckless scientist. He had received training at prestigious institutions and understood CRISPR technology. His stated goal was protecting children from HIV infection by editing a gene called CCR5, which the virus uses to enter immune cells. But the scientific community's response was swift and nearly universal in its condemnation. The twins' father was already infected with HIV, but there were established, effective methods for preventing transmission to children. The genetic edit was unnecessary for their health and safety. More troublingly, the editing was imprecise. The twins didn't receive identical modifications, and there were signs that the edits might not have been clean. He Jiankui had made permanent changes to these children's genetic code without certainty about the consequences. Perhaps most concerning was the irreversible nature of what had been done. These girls would carry their edited genes throughout their lives, and if they have children, approximately half of those children will inherit the modified gene. A decision made by one scientist would affect not just two children, but potentially all of their descendants. The scientific community's response wasn't just about condemning one researcher's actions. It was about establishing clear boundaries around one of the most consequential decisions our species has ever faced. When, if ever, should we make genetic changes that will be passed down through generations? Germline editing means making decisions that will affect people who cannot consent to those decisions, people who may not even be born for generations. This is why scientists worldwide have called for a moratorium on clinical germline editing until we can develop appropriate oversight mechanisms and societal consensus. One of the most profound questions raised by genetic editing is deceptively simple. What is the difference between treating disease and enhancing human capabilities? Where do we draw the line between medicine and improvement? At first glance, the distinction seems clear. When we use CRISPR to restore dystrophin production in boys with muscular dystrophy, that's clearly treatment. When we edit immune cells to better recognize cancer, that's obviously medicine. But as we move away from these clear-cut cases, the line becomes less distinct. Consider genetic variations that don't cause disease directly, but increase the risk of developing common conditions. There are variants that increase the likelihood of heart disease, diabetes, or Alzheimer's disease. If we could edit these risk variants, changing genes that increase heart disease risk from 40 to 20%, would that be treatment or enhancement? We'd be intervening before any disease appears. potentially preventing suffering that might never have occurred anyway. And then there's the possibility of enhancing human capabilities beyond the normal range. What if we could edit genes to increase intelligence, improve memory, enhance physical strength, or provide resistance to aging? Some of these possibilities remain in the realm of science fiction. Intelligence is influenced by thousands of genetic variants, plus countless environmental factors. There's no single smart gene we could edit to reliably increase IQ. But other enhancements might be more straightforward. There are natural genetic variants that provide advantages without causing harm. Some people carry mutations that result in very low cholesterol levels and dramatically reduced risk of heart disease, with no apparent negative effects. If we could give these advantageous variants to everyone, would that be enhancement, or would it be giving all children genetic advantages that some are lucky enough to inherit naturally? Different cultures might answer these questions very differently. Some communities might prioritize genetic changes that enhance physical capabilities. Others might value modifications that affect cognitive abilities. Still, others might prefer to preserve genetic diversity and accept the risks that come with natural variation. Who gets to decide what constitutes an improvement to the human genome? Parents? Medical professionals? Society as a whole? Each approach has merit, but none is perfect. There's a troubling irony at the heart of genetic medicine. The same technologies that promise to eliminate genetic diseases also risk creating new forms of inequality more profound than any our species has ever known. Right now, genetic therapies cost hundreds of thousands of dollars per patient. Some carry price tags exceeding $1 million for a single course of treatment. These costs aren't arbitrary. Developing genetic therapies requires years of research, expensive clinical trials, and sophisticated manufacturing. But the result is that genetic medicine is accessible only to those with extraordinary insurance coverage or personal wealth. Imagine a future where genetic editing becomes routine, where parents can choose to edit their children's genes to prevent disease or enhance capabilities. If these technologies remain expensive and accessible only to the wealthy, we risk creating a world where genetic advantages become hereditary privileges passed down through economically advantaged families. The children of wealthy families might receive genetic edits that make them smarter, stronger, more resistant to disease. The children of poor families would inherit not just economic disadvantage, but genetic disadvantage as well. Over generations, these differences could compound, creating biological castes. This isn't an inevitable future. The history of medical technology shows us that expensive treatments often become more affordable over time. The first antibiotics were luxury medicines available only to the wealthy. Today, they're widely accessible in most parts of the world. The equity challenges become even more complex when we consider global perspectives. Different cultures have different values about genetic modification, different priorities for which conditions should be treated first, different resources available for implementing genetic therapies. There are reasons for hope. International organizations are working to ensure that genetic medicine benefits all of humanity. Scientists are developing CRISPR systems that could be manufactured more easily and administered with less specialized infrastructure. One of the most profound ethical challenges involves a question philosophers have pondered for centuries. How do we make decisions that will affect people who cannot give consent? Tonight, we'll explore how genetic editing is expanding from the realm of human healing into something even more ambitious: the conscious restoration and stewardship of Earth's biological heritage. It's a story that takes us from dying forests to coral reefs under siege, from mosquitoes carrying ancient diseases to microbes living in intimate partnership with our own cells. There's something profoundly hopeful about this expansion of genetic editing beyond our own species. It suggests that the same tools we use to heal genetic diseases in humans might also help us heal some of the damage we've done to the natural world, damage that threatens not just individual species, but entire ecosystems and the intricate relationships that have evolved over millions of years. Let me tell you about a tree that once defined the American landscape, a giant that could grow over 100 feet tall and live for centuries, whose nuts fed both wildlife and early human settlers, whose wood was prized for its strength and resistance to rot. The American chestnut was once called the redwood of the East, dominating forests from Maine to Georgia. But by the 1952, These magnificent trees had virtually disappeared from their native range, victims of a fungal blight accidentally introduced from Asia. Billions of chestnuts died, leaving behind only scattered root systems that could sprout new shoots but couldn't survive long enough to reproduce. For decades, this seemed like a permanent loss, one of the first great extinctions of the modern era. But today, scientists are using genetic editing to write a different ending to this story. They're working to create blight-resistant American chestnuts by carefully editing genes that control the tree's response to fungal infection. The approach is elegantly simple. Researchers have identified genes from other species, including wheat, that produce enzymes capable of neutralizing the toxins produced by the chestnut blight fungus. By introducing these genes into American chestnut cells, they can create trees that retain all their native characteristics, but gain the ability to survive fungal attack. Imagine forests of the future where American chestnuts once again tower over the landscape, their genetic code gently modified to include the molecular tools needed for survival in a world changed by human activity. It's a vision of conservation that goes beyond simply protecting what remains. It's about actively restoring what was lost. But genetic editing for conservation isn't limited to terrestrial environments. In the warming oceans of our changing planet, coral reefs, those underwater rainforests that support a quarter of all marine species, are facing unprecedented challenges from rising temperatures, ocean acidification, and pollution. Some scientists are exploring whether genetic editing might help corals develop greater resilience to these stresses. They're studying coral varieties that have naturally evolved tolerance to warmer waters and trying to understand the genetic basis of this adaptation. The goal isn't to create entirely artificial organisms, but to help existing coral populations acquire the genetic tools they need to survive in rapidly changing conditions. There's something both humbling and inspiring about these efforts. We're using the most advanced molecular tools our species has developed to undo some of the damage our own activities have caused. It's a form of technological penance, an attempt to use human ingenuity to heal wounds we've inflicted on the natural world. Of course, the complexity of intervening in entire ecosystems can't be overstated. Every species exists in intricate relationships with countless others. Changing one organism, even with the best intentions, can have cascading effects that ripple through entire food webs in ways we can't fully predict. But as climate change accelerates and habitat destruction continues, the choice may not be between intervention and non-intervention. It may be between careful, scientifically guided intervention and watching irreplaceable ecosystems collapse. Now, let me share with you one of the most ambitious applications of genetic editing in the natural world, one that could potentially save millions of human lives while raising profound questions about our responsibility for the consequences of releasing genetically modified organisms into wild populations. Malaria, carried by mosquitoes, kills over 400,000 people each year, mostly children in sub-Saharan Africa. For decades, public health efforts have focused on bed nets, insecticides, and anti-malarial drugs. But scientists are now developing a more direct approach, editing the genes of mosquitoes themselves to make them unable to carry the malaria parasite. Every time parents make genetic editing decisions for their future children, they are making permanent changes to another person's genetic code without that person's permission. Of course, parents already make countless decisions that shape their children's lives permanently: where to live, what schools to attend, what traditions to pass along. But genetic editing feels different, more direct in its intervention, more permanent in its consequences. When we edit a child's genes to prevent Huntington's disease, we're changing their fundamental biological nature in ways that cannot be undone. Consider a couple who both carry the gene for Huntington's disease, a devastating neurological condition that appears in middle age. They know there's a 50% chance any child they conceive naturally will develop this disease. If genetic editing could remove the mutation, ensuring their child will never develop the disease, would it be ethical to choose this intervention? Would it be ethical not to choose it? Some disability rights advocates argue that genetic editing based on disease prevention can reinforce harmful attitudes about what kinds of lives are valuable. Many people with genetic conditions live full, meaningful lives despite their diagnoses. Others respond that there's a crucial difference between accepting people who have genetic conditions versus choosing to create new cases when alternatives are available. They argue that preventing genetic disease is an act of compassion, not discrimination. Perhaps the most honest response is to acknowledge the profound difficulty of these questions, while committing to approach them with humility, compassion, and respect for different perspectives. Around the world, different cultures, religions, and philosophical traditions are grappling with genetic editing in ways that reflect their deepest values about what it means to be human. This diversity isn't a problem to be solved, it's one of humanity's greatest strengths. Many religious traditions see the human genome as reflecting divine creation, something sacred that deserves profound respect. The Catholic Church has generally supported somatic gene therapy that treats disease but expressed concerns about germline editing. Islamic scholars have drawn on principles of public interest and the obligation to preserve life, often supporting genetic therapies that prevent disease. Jewish bioethics emphasizes the obligation to heal while respecting human dignity. Indigenous traditions contribute invaluable perspectives on our relationship with genetic inheritance. Many emphasize concepts of kinship that extend to entire ecosystems, viewing genetic diversity as part of the natural world's sacred complexity. Despite their differences, many traditions share common themes: respect for human dignity, concern for future generations, appreciation for life's complexity, and recognition that our growing technological power brings corresponding moral responsibilities. Rather than seeking perfect consensus, the goal is creating space for respectful dialogue, ensuring that genetic editing policies reflect broad input from affected communities, and maintaining appropriate humility about these decisions. The ethical labyrinth we've explored together has no clear exit, no simple path that leads to easy answers. But perhaps that's as it should be. Questions this profound deserve the kind of careful, ongoing consideration that different cultures and traditions can provide. As we've seen, genetic editing raises questions that go far beyond molecular scissors and guide RNAs. They're questions about justice and equity, about consent and autonomy, about human flourishing and the responsibilities that come with unprecedented technological power. The questions we've explored tonight will be with us for generations to come. They deserve our continued attention, our ongoing dialogue, and our commitment to approaching genetic editing with both boldness in pursuing its healing potential and humility about the profound responsibilities it places in human hands. In our next exploration together, we'll expand our view beyond human genetic editing to consider how these same tools are being used to modify other species, to restore ecosystems, create sustainable crops, and address environmental challenges that threaten all life on Earth. The ethical questions don't become simpler, but they do become more hopeful as we explore how genetic editing might help us heal our relationship with the natural world. As you settle deeper into this quiet moment, I want to take you on a journey beyond the boundaries of our own species, into the vast web of life that surrounds and sustains us. The molecular scissors we've explored together, those elegant tools borrowed from ancient bacteria, are not being wielded only in human medicine. They're being used to heal wounded ecosystems, to restore species on the brink of extinction, and to reimagine our relationship with the living world. The technique, called a gene drive, is designed to spread engineered genetic changes through wild mosquito populations. Unlike normal genetic inheritance, where a trait has a 50% chance of being passed to offspring, gene drives can push that probability close to 100%, ensuring that anti-malaria genes spread rapidly through mosquito populations. Think of it as teaching mosquitoes to vaccinate themselves against malaria, then ensuring that this immunity spreads to all their descendants. If successful, it could dramatically reduce malaria transmission, without the need for constant human intervention through pesticides or other control measures. But gene drives also represent something unprecedented in human history: the deliberate, permanent modification of wild species. Once released, these genetic changes could spread across continents, potentially affecting mosquito populations worldwide. The implications are both thrilling and terrifying. Similar genetic editing approaches are being applied to crop plants, but with less controversial implications. Scientists are developing crops that can tolerate drought, resist pests without pesticides, and provide enhanced nutrition. Golden rice, enriched with vitamin A precursors, could prevent blindness in millions of children who lack access to diverse diets. These agricultural applications of genetic editing offer hope for feeding a growing global population while reducing the environmental impact of farming. Crops that need less water, fewer pesticides, and less fertilizer could help us produce food more sustainably. Yet even these beneficial applications raise questions about corporate control of food systems, the rights of farmers to save and replant seeds, and the potential impacts on agricultural biodiversity. The tools are powerful, but their implementation requires careful consideration of social and economic, as well as biological consequences. In the frozen landscapes of Siberia, scientists are working on perhaps the most audacious application of genetic editing ever attempted, bringing back the woolly mammoth. Not through cloning, which would require intact mammoth DNA, but through carefully editing the genome of Asian elephants to recreate mammoth characteristics. The goal isn't to create perfect replicas of extinct mammoths, but to engineer elephants with mammoth-like traits: thick fur, cold tolerance, and the behavioral patterns that once helped maintain Arctic grasslands. These mammophants could potentially help restore tundra ecosystems and slow permafrost melting by recreating the grazing patterns that once kept these landscapes stable. There's something almost mythical about the idea of de-extinction, using genetic editing to undo some of the extinctions caused by human activity. It speaks to our deep desire for redemption, our hope that technology might help us repair some of the damage we've done to the natural world. But the technical challenges remain immense. We're not just editing individual genes, but trying to recreate complex biological and behavioral systems that evolved over millions of years. And even if we succeed technically, there are profound questions about whether bringing back extinct species is ethical or wise. Do we have the right to recreate species that nature allowed to disappear? Do we have the obligation to restore species that human activity drove to extinction? These questions have no easy answers, but they force us to think deeply about our relationship with the natural world and our responsibilities as the first species capable of conscious genetic stewardship. Beyond mammoths, scientists are exploring the possibility of restoring other lost species: passenger pigeons, Carolina parakeets, various plants and animals that have disappeared in recent centuries. Each case raises unique challenges and opportunities, unique questions about what we owe to the species we share this planet with. As we near the end of our journey tonight, I want to bring us back to something much closer to home indeed. Something that is literally part of home. Right now, as you rest there, your body hosts trillions of microorganisms, bacteria, archaea, fungi, and viruses that live in intimate partnership with your human cells. These microbes aren't just passengers, they're active participants in your health and well-being. They help digest your food, synthesize vitamins, train your immune system, and even influence your mood and behavior through the gut-brain connection. In many ways, you are not a single organism but a walking ecosystem, a collaborative community of life forms working together. Scientists are now beginning to explore how genetic editing might be used to modify these microbial partners, not to eliminate them, but to enhance their beneficial functions or restore healthy microbial communities that have been disrupted by antibiotics, poor diet, or other factors. Imagine being able to edit the genes of your gut bacteria to help them produce more of the neurotransmitters that support mental health, or to enhance their ability to break down harmful compounds before they can cause inflammation. Imagine restoring the microbial diversity that our ancestors enjoyed before the modern disruptions of industrial life. The microbiome represents a frontier where genetic editing blurs the boundaries between self and other, between human medicine and environmental science. When we edit our microbial partners, are we editing ourselves? Are we restoring natural relationships or creating artificial ones? These questions remind us that genetic editing isn't just about individual organisms, it's about relationships, partnerships, and the complex webs of interaction that make life possible. Whether we're restoring forests, protecting coral reefs, or nurturing the microscopic gardens within our own bodies, genetic editing is becoming a tool for conscious participation in the ongoing evolution of life on Earth. The molecular scissors we've explored together, those elegant tools borrowed from ancient bacterial immune systems, are opening possibilities that extend far beyond human medicine. They're becoming instruments of ecological restoration, agricultural innovation, and even planetary stewardship. As we develop these capabilities, we're not just gaining power over individual genes or organisms. We're becoming conscious participants in the evolutionary process itself, with the ability to guide the development of life in directions we think will be beneficial for our planet and its inhabitants. The responsibility is enormous, but so is the potential. The same tools that can cure genetic diseases in humans might also help us restore damaged ecosystems, feed growing populations sustainably, and create new forms of partnership between human technology and the natural world. In our final exploration together, we'll look toward the future, examining the most cutting-edge developments in genetic editing and imagining how these tools might continue to evolve, what new possibilities they might open, and how they might reshape our understanding of what it means to be alive in an age when the code of life itself has become editable. For now, rest in the wonder of living in a time when human ingenuity is beginning to serve not just our own species, but the entire community of life on Earth. The molecular tools we've developed are becoming instruments of healing not just for individuals, but for ecosystems, species, and the intricate relationships that connect all living things. As we settle into the deeper hours of our exploration together, I want to share with you something that scientists often whisper about in quiet moments: the humbling reality that even our most elegant tools, our most precise interventions, sometimes create music we didn't intend to compose. Tonight, we'll explore the unintended symphony of genetic editing, the off-key notes, the unexpected harmonies, and the beautiful complexity that emerges when we intervene in biological systems that have been composing themselves for billions of years. This isn't a chapter about failures or fears, but about the profound mystery that remains at the heart of life, even as we develop tools that can edit individual letters in the genetic alphabet. It's a reminder that biology is not a machine we can control with perfect precision, but a living symphony where every note we change can create ripples of sound we never anticipated. There's something both humbling and beautiful about acknowledging these limitations. They remind us that genetic editing, powerful as it has become, is still a conversation between human intention and biological reality, a conversation where biology sometimes has the final word. Imagine you're a master calligrapher, working with the finest brush, attempting to correct a single character in an ancient manuscript. Your hand is steady, your intention is clear, but sometimes, just sometimes, a tiny drop of ink falls where you didn't intend it to fall, creating a mark you never meant to make. This is what geneticists call an off-target effect, when our molecular scissors, guided by their RNA addresses, occasionally cut at locations in the genome that we never intended to edit. Despite the extraordinary precision of CRISPR and related tools, they are not perfect. Sometimes they find sequences that look very similar to their intended target and make cuts there as well. The reality is both more complex and less alarming than it might first appear. Most off-target cuts occur in regions of the genome that don't code for proteins in those vast stretches of DNA that we once called junk, but now understand to contain regulatory elements and other important information. Many off-target edits have no detectable effect on cell function, but some do, and this is why genetic editing researchers spend enormous amounts of time and effort trying to predict, detect, and minimize these unintended changes. They've developed increasingly sophisticated methods for mapping exactly where their molecular scissors cut, for designing guide RNAs that are more specific, and for creating editing tools that are less likely to make mistakes. There's also the phenomenon scientists call mosaicism, when genetic editing treatments don't reach every cell they're intended to modify. Imagine trying to paint every leaf on a vast tree, but your brush only reaches some of them. The result is a mixed population of cells, some that carry the intended genetic edit, others that remain unchanged. In early gene therapy trials, researchers sometimes found that only 30% or 40% of target cells had been successfully edited. This partial editing can still provide therapeutic benefits often. You don't need to fix every cell to restore function, but it adds another layer of complexity to treatment outcomes. The encouraging news is that these tools continue to improve with remarkable speed. Scientists have developed base editors and prime editors that make fewer off-target cuts. They've created delivery systems that reach target cells more efficiently. They've refined their methods for predicting and avoiding potential problems. But perhaps most importantly, they've learned to approach genetic editing with appropriate humility, understanding that uncertainty is not a failure of the technology, but an inevitable aspect of pioneering medicine. Every early patient who volunteers for genetic editing trials is helping to map the boundaries between intention and reality, contributing to our understanding of how to make these treatments safer and more effective. There's a beautiful irony at the heart of genetic medicine that might make you smile. even in this quiet hour. The same immune system that we're sometimes trying to enhance with genetic editing can also recognize our editing tools as foreign invaders and mount a response against them. Our immune systems have evolved over millions of years to be suspicious of anything that looks foreign, anything that shouldn't be there. When we introduce CRISPR proteins, base editors, or viral vectors carrying genetic cargo into the body, our immune cells sometimes sound the alarm. Unknown molecular machinery detected. Potential threat. It's like trying to help repair someone's house while their very protective guard dog keeps barking at your tools, mistaking your helpful intentions for a burglary in progress. The immune response isn't wrong, it's doing exactly what it evolved to do. But it can interfere with the very treatments we're trying to deliver. This immune recognition can happen at multiple levels. Sometimes the body recognizes the delivery vehicles, the modified viruses, or lipid nanoparticles that carry genetic editing tools to their targets. Other times, it responds to the editing proteins themselves, especially if they're derived from bacterial sources like the original CAs9. The consequences can range from reduced effectiveness of the treatment to more serious inflammatory responses. In some early gene therapy trials, patients developed immune responses that not only blocked the intended therapy, but also prevented them from receiving additional treatments in the future. But scientists are learning to work with our immune systems rather than against them. They're developing ways to temporarily suppress immune responses during treatment, creating more stealth delivery vehicles that evade immune detection, and even engineering editing tools that look more like human proteins and less like foreign invaders. There's something profound about this ongoing conversation between our intentions and our biology. Our immune systems, in their protective vigilance, are teaching us to be better genetic editors, forcing us to develop tools that work in harmony with our natural defenses rather than triggering them. It's a reminder that genetic editing isn't just about the mechanical precision of molecular scissors, but about the delicate art of working within living systems that have their own intelligence, their own priorities, their own ways of maintaining the balance that keeps us healthy. In chaos theory, there's a beautiful concept called the butterfly effect, the idea that a butterfly flapping its wings in one part of the world might, through a chain of increasingly complex interactions, influence weather patterns on the other side of the planet. In genetic editing, we encounter something similar. The recognition that changing even a single gene can sometimes create ripples of effect that spread through biological systems in ways we never anticipated. Every gene exists in a web of relationships with other genes, other proteins, other cellular processes. When we edit one component of this network, we're not just making an isolated change, we're potentially affecting the entire system's dynamics. Consider the seemingly simple goal of editing a gene to reduce cholesterol production. This sounds straightforward, but cholesterol isn't just a potential problem for heart health. It's also a crucial component of cell membranes and a precursor to important hormones. Reduce it too much, and you might inadvertently affect cellular function or hormone balance in ways that create new problems. These ripple effects become even more complex when we consider genetic editing in wild populations. The mosquitoes we engineer to resist malaria might also have altered behaviors that affect their relationships with other species. The crops we modify for improved nutrition might interact differently with soil microbes or insect pollinators. This isn't an argument against genetic editing, but rather a call for the kind of humility that complex systems demand. When we intervene in networks that have been evolving for millions of years, we must expect the unexpected, prepare for surprises, and remain ready to learn from outcomes we didn't predict. The butterfly effect in genetics teaches us that courage and caution are not opposites but partners. We need the courage to pursue genetic editing's healing potential, coupled with the caution to proceed thoughtfully, monitoring for unintended consequences and remaining ready to adjust our approach as we learn more. As we near the end of our exploration tonight, I want to share with you one of the most profound aspects of genetic editing, what former U.S. Secretary of Defense Donald Rumsfeld famously called the unknown unknowns, the things we don't know that we don't know. In the early days of genetic editing, researchers focused primarily on what they called known unknowns, questions they knew they needed to answer. How precise could they make their tools? How efficiently could they deliver them to target cells? What were the most common off-target effects? But as genetic editing moved from laboratory experiments to clinical trials, Scientists began encountering surprises they hadn't anticipated, biological responses they hadn't predicted, therapeutic effects they hadn't expected, and sometimes challenges they hadn't imagined. Some patients responded to genetic editing treatments in ways that exceeded the most optimistic predictions. Others showed responses that were beneficial, but completely different from what researchers had expected. Still others taught scientists about biological pathways and cellular mechanisms that hadn't been considered in the original treatment design. These surprises aren't failures of scientific planning, they're inevitable features of working at the frontier of human knowledge. Every unknown that genetic editing reveals teaches us something new about biology, about disease, about the intricate systems that maintain life. The patients who volunteer for early genetic editing trials are not just receiving experimental treatments, they're serving as pioneers, helping to map the territory between what we think we know and what biology actually does. Their courage makes possible the safer, more effective treatments that will benefit future patients. There's something beautiful about acknowledging the limits of our knowledge while still pursuing the possibilities that genetic editing offers. Science has always been a conversation with mystery, a careful dance between human curiosity and natural complexity. The unknown unknowns of genetic editing remind us that we are still students in the vast classroom of life, still learning how to read the genetic text that has been writing itself for billions of years. Our molecular scissors are remarkable tools, but they are tools wielded by beings who are still discovering what it means to edit the code of life responsibly. The unintended symphony we've explored tonight, the off-target effects, the immune responses, the ecological ripples, and the unknown unknowns is not a discordant noise, but rather the natural music that emerges when human intention meets biological reality. Sweet dreams, and may your rest be filled with the quiet wonder of being alive in an age when the code of life itself has become a text we can read, understand, and gently edit in service of healing and hope. Each unexpected note teaches us something new, helps us compose future interventions more thoughtfully, and reminds us that genetic editing is not about imposing human will on passive biological systems, but about learning to collaborate with the living world in ways that honor both our healing intentions and the deep wisdom embedded in life itself. As we prepare for our final exploration together, let us rest in the knowledge that uncertainty is not the enemy of progress, but its essential companion. The mysteries we encounter in genetic editing are not obstacles to overcome, but invitations to deeper understanding, opportunities to develop not just more powerful tools, but greater wisdom about how to use them. The symphony continues, with movements we can compose and movements that will surprise us, all part of the great musical conversation between human knowledge and the enduring mystery of life. As dawn approaches in our journey together, let us gaze toward horizons where genetic editing continues to expand its gentle reach into the mysteries of human existence. Scientists are learning to grow organs from a patient's own cells, potentially ending the heartbreak of transplant waiting lists. Imagine hearts, livers, and kidneys cultivated in laboratories, genetically matched to their recipients. Some researchers are even editing pig organs to be compatible with human biology xenotransplantation that transforms other species into living pharmacies for human healing. The molecular clocks that govern aging are yielding their secrets to genetic editing. We're discovering how to help cells resist the accumulated damage of time, potentially extending not just lifespan, but health span. Yet this raises profound questions. How long should humans live? The goal isn't just adding years to life, but adding vitality to years. The genetic threads woven through depression, anxiety, and cognitive differences are being carefully mapped. While the complexity of consciousness remains beautifully mysterious, genetic editing may offer new approaches to mental wellness, not changing who we are, but helping us become our most balanced selves. The future promises medicine as individual as fingerprints, where each person's unique genetic profile guides their treatment. Our genes will tell doctors which medications work best, which doses are optimal, creating therapies tailored with unprecedented precision. As our journey through the landscape of genetic editing draws to a gentle close, the first light of understanding illuminates the horizon of what we've discovered together. We began this exploration with a simple question echoing through the quiet darkness. What happens when we edit the code of life? Now, in this contemplative hour, let us gather the threads of wonder we've woven and see what tapestry of meaning emerges. The question that opened our conversation feels different now, doesn't it? What seemed like a straightforward inquiry has revealed itself to be as layered and complex as the genetic code itself, beautiful in its intricacy, profound in its implications, and still being written with each passing day in laboratories and clinics around the world. So what happens when we edit the code of life? The answer, as we've discovered together, is not a single note, but an entire symphony complex, beautiful, sometimes discordant, and always still being composed. We gain the power to heal in ways that would have seemed like magic to previous generations. We can correct the single-letter typos that cause sickle cell disease, restore sight to those born blind, teach immune systems to recognize and destroy cancer cells, and potentially give strength back to muscles weakened by genetic disorders. We can reach into the very instruction manual of life and make gentle corrections that transform suffering into hope. But with this extraordinary power comes equally extraordinary responsibility. We've learned that genetic editing is not just a technical challenge, but a profound moral one. Every edit we make carries weight not just for the individual receiving treatment, but potentially for their children, their communities, and our species as a whole. We gain the responsibility to use these tools wisely, ensuring that genetic medicine benefits not just the privileged few but all of humanity. We bear the weight of making decisions that will echo through generations, of balancing individual autonomy with collective well-being, of proceeding with both courage and caution as we navigate ethical territories that no generation before us has ever had to explore. The power to edit genes reminds us that we are not separate from the biological systems we seek to heal, but intimately connected to them through bonds forged over billions of years of shared evolutionary history. In learning to edit the code of life, we are discovering that we ourselves are living expressions of that same code, temporary arrangements of molecules animated by the ancient dance of DNA, RNA, and protein. We also gain something perhaps more precious than the ability to heal specific diseases, a deeper appreciation for the extraordinary complexity and beauty of living systems. Every successful genetic therapy is a testament not just to human ingenuity, but to the remarkable cooperative capacity of our cells, which somehow recognize and integrate our molecular interventions into their ongoing work of maintaining life. One of the most profound realizations from our exploration together is how genetic editing reveals the fundamental unity underlying all life on Earth. The same four-letter alphabet that spells out the instructions for human consciousness also guides the growth of ancient trees, the flight patterns of migrating birds, and the photosynthetic machinery that converts sunlight into the energy that sustains our planet's ecosystems. When we use CRISPR tools borrowed from bacterial immune systems to heal human genetic diseases, we are participating in a conversation that has been ongoing for billions of years, a molecular dialogue between different forms of life, sharing genetic innovations across species boundaries, collaborating in the grand project of biological existence. This shared genetic heritage means that editing human genes is not just an intervention in human biology, but a conscious participation in the continuing evolution of life itself. We are not outside observers manipulating biological systems like machines, but conscious participants in those systems, using our awareness and intentionality to guide processes that have traditionally been left to the unconscious wisdom of natural selection. There's something deeply humbling about recognizing our place in this vast genetic library that encompasses all living things. We are neither the authors of the genetic code nor separate from it, but rather conscious readers who have learned, for the first time in Earth's history, to make deliberate edits in the text of life itself. This recognition brings with it a sense of kinship with all living things, a molecular reminder that the boundaries between species, while real and important, are also permeable and interconnected. The genetic editing tools we use to heal human diseases work because life shares common molecular mechanisms across vast evolutionary distances. As we draw toward the end of our time together, I want to pause in gratitude for the countless individuals whose courage, curiosity, and dedication make genetic editing possible. Behind every story of healing we've shared tonight are patients who volunteered to be among the first to try experimental treatments, knowing that their experiences would pave the way for others, but offering no guarantee of personal benefit. There's Victoria Gray, living free from sickle cell pain because she was brave enough to have her genes edited with technology that had never been tried in humans before. There are children in clinical trials for inherited blindness, hoping that genetic editing might restore sight while knowing they might be helping researchers learn more than they are personally helped. Behind these patients are researchers who dedicate their careers to understanding the molecular mechanisms of disease and healing, scientists who spend years developing safer delivery systems, more precise editing tools, and better methods for ensuring that genetic therapies reach the cells that need them most. There are clinicians who conduct these trials with extraordinary care, monitoring patients for both beneficial effects and potential complications, learning from each case how to make genetic editing safer and more effective for future patients. And there are the countless laboratory technicians, regulatory scientists, ethics committee members, and advocates who work behind the scenes to ensure that genetic editing advances in ways that are both scientifically rigorous and ethically responsible. We are living through a remarkable moment in human history, perhaps the most significant medical revolution since the discovery of antibiotics. The fact that we can now edit the genetic instructions that govern life with precision and safety, that improve every year, is a testament to human ingenuity, perseverance, and our deep commitment to healing. There's something almost magical about the realization that the molecular scissors we use for genetic editing were originally developed by bacteria to defend against viral infections, that life itself provided us with the tools we needed to become conscious genetic editors, even though evolution had no knowledge of our future needs. As we gain the power to edit life's code with increasing precision and scope, something beautiful is happening to our relationship with the genetic instructions we inherited from billions of years of evolutionary history. We're learning to read them not just as technical specifications for biological function, but as poetry, ancient verses written in molecules, telling the story of life's long journey, from simple chemical reactions to conscious beings capable of contemplating their own existence. Every successful genetic therapy represents a collaboration between human intention and cellular wisdom. When we edit genes to treat sickle cell disease, we provide the molecular instructions, but the patient's cells do the actual work of implementing those instructions, translating genetic changes into healthier blood cells, integrating new genetic information into their ongoing biological processes. This collaboration reveals something profound about the nature of healing itself. Genetic editing succeeds not because we can impose our will on passive biological systems, but because we've learned to work in partnership with the inherent wisdom of living cells, cells that still remember, at a molecular level, how to maintain health, how to repair damage, how to adapt to new circumstances. In editing the code of life, we're not just changing DNA sequences, we're participating in the ongoing story of what it means to be human. We're using our uniquely human capacities for conscious choice and technological innovation to expand the range of human possibilities, to reduce suffering, and to enhance our ability to live full, healthy lives. But we're also discovering that our power to edit genes comes with the responsibility to exercise that power wisely, with respect for the complexity of biological systems, with concern for justice and equity, and with humility about the limits of our understanding. Perhaps most importantly, genetic editing is teaching us that we are not separate from the natural world, but intimately connected to it through the molecular mechanisms we share with all living things. In learning to edit the code of life, we're not transcending our biological nature, but embracing it more fully, becoming conscious participants in the evolutionary process that gave rise to us. As our exploration draws to its peaceful close, I invite you to rest in the knowledge that even as you sleep, the patient work of science continues. In laboratories around the world, researchers are refining genetic editing tools, conducting clinical trials, and developing new approaches to healing that were unimaginable just a few years ago. Thank you for sharing this journey of discovery with me, for bringing your curiosity and wonder to these profound questions about genetic editing and what it means for our future. There's something beautiful about learning together, about sharing the excitement of scientific discovery and the deeper questions it raises about our place in the universe. In your genes, you carry the stories of every ancestor who survived long enough to pass their genetic inheritance forward to you. You also carry the possibilities of every descendant who might someday benefit from the genetic editing technologies we're developing today. You are both the product of evolution's long journey and a conscious participant in directing where that journey goes next. The genetic code you carry is written in the same four-letter alphabet that guides every form of life on Earth, the same molecular language that shapes the growth of forests, the migration patterns of whales, and the complex ecosystems that support our planet's incredible biodiversity. Sleep well, fellow travelers in this cosmic genetic library. Tomorrow, new chapters await new discoveries about the code of life, new possibilities for healing, new questions about how we should use our growing power to edit the instructions that make us who we are. Rest knowing that the story of genetic editing is still being written, and that each of us, in our own way, is helping to write it. Dream of molecular scissors that cut with precision and compassion, of genetic therapies that heal without harm, and of a future where the power to edit life's code serves the flourishing of all living things.